Very welcome to this session. It's quite a diverse session, I think, on the various topics that are very interesting to us as food microbiologists. The first presentation, but I don't see the presenter just yet, is by Lerato Mogozzi, and she's from Bloemfontein. I hope she's in the room now and can start. Yes, there you are. So the floor is all yours.
Thank you. Good day, ladies and gentlemen. As being mentioned, I'm Lerato Mogozzi. I'm from Central University of Technology in Bloemfontein, South Africa. And the research center that I come from is Center of Applied Food Sustainability and Biotechnology. So the title of my talk this morning is Microbial and Associated Contamination in South African Craft Ales. Beer is the most consumed alcoholic beverage worldwide, followed by tea and coffee, as well as water. Czech Republic is a country that consumes most beer. However, South Africa is not the biggest drinking nation in the African continent, with about 56% consumption coming from beer. Contemporary trend of focusing on micro-manufactured produce, also known as a craft beer development, is known as a microbrewer movement as well. Craft beer, as compared to industrial beer, is more prone or subject to microbial contamination. This is because most of the craft breweries don't have resources in place to pasteurize as well as filter their beers. So this results in the proliferation of gram-negative bacteria as well as wild yeast. However, majority of spoilage incidents, they come from lactic acid bacteria. Microbial contamination can affect the beer quality, and this can result in disastrous financial consequences in the microbreweries. So this microbial contamination can be associated with mycotoxins that come from molds, as well as off flavors that can be associated with lactic acid bacteria. So most of the craft brewers, as I've mentioned, they don't have resources in place. Most of their expenditure, it goes to the following. 4% can go to the brewery margin, packaging 18%, 15% to labor, 9% raw materials, 23% due to license fees, and 11% distribution, and 20% to retailer margin. Mycotoxins are secondary metabolites of fungi that can be associated with cereals such as your barley, your maize, as well as your wheat. These are thermally and chemically stable compounds that can contaminate the finished processed foods such as your beer. Mycotoxins, as a result, can cause health disorders when ingested in high concentrations. So in order for us to understand how these off flavors end in the beer, we first have to understand how they come about in the beer, as well as their metabolism as well. So in the beer, we have what you call hydrocyanamic acids, like your p-comoric acid and ferulic acid. Those are two abundant hydrocyanamic acids that can be found in the beer that can be extracted from your grain during brewing. So this hydrocyanamic acid, your comoric acid and ferulic acid, normally they come in high threshold concentrations, in your aroma and your flavor. That can affect your aroma and your flavor as well. So these hydrocyanamic acids, they can be decarboxylated to your vinyl products as well as your ethyl products. So your vinyl products and your ethyl products, they normally come in low threshold concentration in your beer. However, their presence can become, they're not very likable to the drinker as well. They're not very good in terms of their presence in the beer as well. So they're not acceptable most of the times. They shouldn't be found there. So sometimes they can be associated with some form of contamination as a result of an enzyme called phenolic acid decarboxylase. So if there's a presence of a contaminant in beer that can decarboxylate this phenolic acid, that can decarboxylate these hydrocyanamic acids, it's not good also in terms of the beer quality. Thus, the aim of our study was to determine the level of microbial contamination in South African craft ales as well as to detect any associated compounds that could prove hazardous to consumer health or influence the flavor profile. Now, these are the materials and methods that we followed. We sampled different ale beers in terms of the different styles. But in brewing, we have ale beers. We also have what you call lager beers. So we actually focus on ale beers. So we sampled different kinds and types of ale beers from 30 different microbreweries around South Africa. So if you can look at the map on your right, this is the map of South Africa. We've got about 9 provinces. So we sampled different beers from those different provinces. But most of our craft breweries are mainly located in the Western Cape. The Western Cape. So that's where most of our beers were actually bought from. And the different kinds of types of beers that we sampled, that are ales, as I've mentioned. We have your IPAs, your English ale, your stout, your wheat beer, imperial porter, blonde ale, American pale ale, and amber ale. So once we got these samples from these different provinces, we stored them at 4 degrees Celsius until further analysis. So the analysis that were done, we actually were looking for pathogens that can be associated with the beer. We looked also at the spoilage bacteria as well as the yeast, mold and yeast. And we used different selective media in order to be able to detect them. We also took about 50 mL of both for mycotoxins as well as volatile compounds. So our mycotoxins of interest were deoxynivalenol, aflatoxin, nivalenol, fumonisin, ochratoxin, and zearalenol. So according to literature, deoxynivalenol, nivalenol, and as well as fumonisin are more common in your grain that we use for brewing. The volatile compounds that can be associated with contamination and that are normally good for sensory analysis or that are not good for their presence in a beer. We looked at 4-vinyl guaiacol, 4-ethyl guaiacol, 4-vinyl phenol, and 4-ethyl phenol to see if there's any association between them and lactic acid bacteria. So according to our results, this is for yeast and mold contamination in craft ale beers. For our beers, IPA, which is Indian Pale Ale, we only found about 45 coliform units per ml. So this is how we actually graphed this. On my right, we have different styles of beers. So we sampled from 30 different breweries, but we actually grouped them according to their styles to see which beer contained how much of the coliform units. And we saw that IPA, we have the highest of 45, and your stout is 28.3. So most of the beers didn't have any yeast and malt contamination, which is quite a good thing. And we couldn't even associate the presence of this yeast and mold with any of the mycotoxins. So for lactic acid bacteria, as well as other microorganisms as well that I've mentioned, your pathogens as well, we didn't detect any pathogens. We're talking about your coliforms, we didn't detect any of those. Your salmonella, we didn't detect any of those. But we actually found your lactic acid bacteria, which is also very interesting according to literature because it's very resistant to intrinsic conditions of the beer. So in this, the range of what we actually saw here was between 20 to 177,500 coliform units per ml of lactic acid bacteria, which means 63% of all the craft beers that we actually looked at were contaminated with lactic acid bacteria. Mycotoxins, according to literature, thus far, we don't have any maximum tolerable doses of mycotoxins that has been found in beer. In the grain, like the barley, yes, we have. But according to the literature that we went through and our studies, there was no significant amount of mycotoxins that should be of concern that was detected in the beers that we actually sampled. Now, we also wanted to look at the off flavors, as I've mentioned. Everything that you see highlighted in red, it shows that all these concentrations in these different types of beers were beyond or above the threshold limit. So if you can see on my table above, everything that is highlighted in black in those blocks above, it just show you the the accepted threshold that is needed to be in those different beers. And in the first column, we have 4 vinyl guaiacol. You can see that in most of them, they have this specific compound as well. So also we wanted to see if there's any correlations at all between those off flavors and lactic acid bacteria, because it's known that lactic acid bacteria can produce a specific enzyme that can result in these off flavors. But not only lactic acid bacteria, other organisms as well, like your wild yeast, can have this specific enzyme. But according to spear correlations that we did here, we saw that there was no correlation at all between lactic acid bacteria and those 4 off flavors, that is 4-ethyl guaiacol, 4-ethyl phenol, 4-vinyl guaiacol, and 4-vinyl phenol, as well as vanillin. So there's absolutely no correlation. So everything that went beyond 0.5, we saw it as a correlation. Anything beyond the coefficient of 0.5, then there was no correlation at all, as you can see. Now to conclude, we say microbial quality of South African craft ales needs attention due to the fact that 63% of all our craft beers were contaminated with lactic acid bacteria. So quality assurance, not only of the final product, is vital in our craft beer industries, but the raw materials as well, which can carry mycotoxins, is crucial up until throughout the craft brewing process. And according to literature, mycotoxin levels are within the allowed limits. And also we found that there's totally no correlation between this lactic acid bacterial contamination to the off flavors that we detected in the beer. Now, for our future research, we need to do the next generation sequencing in order to understand the lab community, lactic acid bacterial community, as well as detect the presence of wild yeast, because we couldn't understand what caused those high levels of off flavors. So taking another step to do a next generation sequencing, we revealed if there's any wild yeast that we can associate with these off flavors. And also our future plan is also to look at other chemical compounds that can be associated with any form of contamination, yes, like your major volatiles, for example, your esters, and look at the sugar profile as well. Thank you.
Thank you very much. Are there any questions from the audience? Okay, let's see it.
Thank you for such a wonderful presentation, and I'm from Hungary. I'm interested, I want to ask 2 questions because you were talking about lactic acid bacteria. Can you just specify a little bit which group of lactic acid bacteria was more prevalent? And you said that there was no yeast present, right?
No.
Yeah, but
wild yeast.
I mean, I don't know much about the craft beer, but all I know is that the yeast actually gives a flavor to the craft beer. So why was this difference?
Okay.
From your observation and that.
All right, thank you. I was actually referring to the wild yeast. The wild yeast is the yeast that's not supposed to be there. We have the brewery yeast and the wild yeast. The wild yeast is a contaminant that can come through maybe when you're inoculating your beer or during the brewing process. That's the wild yeast that I was talking about. And then your first question referred to, sorry? Oh, we haven't done the profile yet. That's the next step that we need to do in a study to see what type of lactic acid bacteria that we need to, that we're talking about here. Yeah.
Okay. Thank you very much for your presentation. If there's no further questions. So the next presenter will be the co-chair of today, and this is Aspasia Nisiotou, and she works here in Athens at the Elgo Dimitra.
Thank you very much. Good afternoon, everyone. In this presentation, we will show our work on the development of novel molecular markers based on microsatellite analysis for genotypic Metschnikovia pulcherrima, which is a common wine-related species, but hasn't received much scientific attention till now. Let me start with some information about wine quality and yeasts. Wine, we all know that is among the most valuable and prized agricultural commodities. It is a huge market that is projected to grow even more within the next years. And the top drivers of its value include the vintage quality, the provenance, the rarity, and the ability to age. It is a highly complex product that contains more than 1,000 chemical constituents, which originate from the grape vineyard, the specific grape vineyard that is used to make the must, and of course, a microbial activity. The close connection between the wine quality and the place of origin is summarized into the term vitivinicultural terroir. This term describes the influence of the place of origin in the special features of a specific wine. Traditionally, typically, the main components of the terroir in wine include the climate of the region, the soil, the terrain, that includes the altitude, the slope, and other characteristics, and the tradition that include the vitivinicultural practices that are in this region. Of course, the question that the food microbiology asks is what is the role of the native microbiota? Has any role in the terroir notion? If that is the case, then the term microbial terroir emerges and implies that, hypothesizes that the geographically distinct viticultural regions may possess different yeast communities. That means that the first criterion to be met for microbial terroir to exist is that the wine-associated microbiota exhibits geographical patterns, geographical distributions. This is not enough. The second criterion that has to be met is that this microbial geographical differentiation results in metabolic differentiation that is translated to phenotypic differentiation in wine. This is a critical factor in consumer preference and economic appreciation and needs to be confirmed. So which microorganisms can be included in this notion? Of course, the microorganisms that have a significant metabolic imprint in the wine product, and that are typically the fermentative yeasts and the lactic acid bacteria that conduct the alcoholic and malolactic fermentation, respectively. We focus more on yeast due to their decisive role in the alcoholic fermentation. We can divide the wine-related yeast in two major groups. One is the Saccharomyces cerevisiae, of course, the prime wine yeasts that conduct the alcoholic fermentation with its numerous strains. And the other group includes some diverse yeast species that have different metabolic activities compared to cerevisiae and to each other. With these ideas in mind, we started back a few years ago a large-scale sampling. We screened the central and the southern Greece, the main protected designation of origin regions in Greece, 42 vineyards, 3 sampling points per vineyard, and we followed the sampling for 2 or 3 years when grapes were available. We collected more than 2,500 yeast isolates, among which 500 belonged to Metschnikovia pulcherrima. So, of course, the first step was to check whether there was any geographical regionalization of the yeast communities at the species level. And we performed several statistical analyses and found that species composition of yeast communities actually differed significantly among the different regions. This is depicted in the three diagrams, as can you see, where the yeast community is depicted by the dots, grouped together when they belong to a specific region, as shown by different colors. The next observation was the high abundancy of Metschnikovia pulcherrima, which was, along with Hasseniaspora ovarum, the most prevailing species in the Greek vineyards. So we decided to study more this species because it seems to be important, since it has some important metabolic activities. But due to its low to moderate fermentation power, was not considered till now for technological applications. Among the other metabolic activities, the production of hydrolytic enzymes that influence the sensory character of wines, and then the antimicrobial activity due to the production of pulcherrimic acid is some of them that show specific interest. Other activities important for improving the wine aroma are the production of phenylethanol, of several esters like isoamyl acetate, activity on terpenes, and these all characteristics have been found to be largely strain dependent, as often happens in yeast species. So we couldn't find any dedicated tools for typing our isolates, so we consider to do genotyping by microsatellite analysis. This method, microsatellites or simple sequence repeats, are tandem repeated DNA sequences of normally 1 to 10 BP. They are highly polymorphic because they are prone to errors during DNA replication, and they show high reproducibility among laboratories. So in the figures, you can see an example of repeat units consisting of 1, 2, 3, up to 6 nucleotides, and the common practice is to amplify these regions with primers and then detect the differences in the length polymorphisms in a genetic analyzer. To do this for Metschnikovia pulcherrima, we use the genome sequence of strain APC 1.2 that is available at NCBI. And we analyzed for microsatellites of 2 or more nucleotides using the tandem repeat finder program. Only microsatellites of at least 10 repeats were selected in order to increase the possibility for them to be polymorphic. And we designed primers that flank the microsatellite regions, and we use them in amplification experiments. This is a table showing the 15 microsatellite loci that were selected within the gene of Metschnikovia pulcherrima and retained for further analysis. You can see the nucleotide motif that is, ranges from 2 to 4 nucleotides, the nucleotide motif, the chromosome that is located, and the specific primers that were designed flanking the microsatellite region. Of course, then these microsatellites had to be tested. We used a panel of indigenous Metschnikovia pulcherrima strains along with the type strains to test the efficiency and the differences in the sizes. And we select the 9 ones, which you can see the highlighted ones, to perform the analysis in the total number of isolates that we have in our lab. 9 of them were found to show allele variation. They were located at 4 different chromosomes. The strains were first typed with a generic method for Ascomycetes to have groups of isolates, and then we applied them to 66 vineyards isolates from the different regions. So as we speak, the genetic analyzer is running. So for the future goals, I will show you an example of a similar analysis that we have performed in our lab. And what we want to do, we want to take the fragment lengths and analyze them, so to find the genetic diversity between Metsnikovia pulcherrima isolates from the different regions. And this is an example from a similar experiment that was conducted on Laissezia thermotolerans, another wine-related yeast that is well known for its ability to do biological acidification in wine, with very relevance to climate change in winemaking. So in this case, we found that the genetic analysis of thermotolerance based on microsatellites showed a clear structure between the populations of isolates originating from two different regions, from Nemea and Pezao, in PDO regions in Greece. The next step is to evaluate the metabolic profile, the metabolic activity of these isolates, of the different isolates, with the purpose to compare the metabolic profiles between the strains from different regions, from different vintages, from different vineyards, and to draw some results. And this is, again, an example from Laissezia thermotolerance that was discriminated based on kinetic and phenotypic profiles according to the regions they originate. Why would we do this? Of course, these have significant applications in winemaking. We can succeed in differentiating wines to give special characters. We can introduce a terroir effect in wines. We can use strains, specialty yeasts to ferment difficult masts, to increase the volatile profile of the wine or decrease any specific faults, and of course to adapt to the recently emerging climate change. And this is an example of another experiment performed in our lab, where we used indigenous strains to differentiate the producing wine. Thank you very much, and I'm happy to answer any questions you may have.
So I'd like to invite people in the audience if anyone has a question. Yeah.
Hi, thank you for the talk. We used, like 10 years ago, SSR also to identify ploidity levels. So if you have a triploid or tetraploid, you see a few alleles on the same strain. Depends if it is homothallic or heterothallic. So.
Yes. This is why we selected microsatellites also to check the ploidy of the yeast, because it's very important, especially in wine strains, because they show high level of ploidy. And this is an advantage of this method for yeast isolates. Thank you for your comment.
Okay, in view of time, I would like to ask anyone who has further questions, perhaps to do that after the lecture.
Thank you very much.
So, thank you. Then the next lecture will be online via Zoom. So this will be presented by, first I have to change glasses here, sorry. Presented by Raquel Nogueira, and she works at the Marine Research Institute in Vigo. So I hope the connection is working. Yes, hello, welcome.
Hello, thank you. I'm sharing my screen. Do you see it?
Yes, perfect.
Yes.
Okay.
Thank you. Go ahead.
Good afternoon, everyone. I'm Raquel Nogueira, and I'm going to present to you the principal component analysis as a tool to cluster Listeria monocytogenes isolated from food-related environments based on their physiological and three-dimensional biofilm features. As you know, Listeria, as foodborne pathogen rose a worldwide concern due to his constantly outbreaks. In 2020, based on the EFSA information, were reported 1,876 cases of listeriosis and 167 deaths within the European Union. Listeria biological features allow it to thrive and survive and thrive in food and food processing environments. And regarding risk assessment, different classifications have risen along the years to sort Listeria according to one or another biological features. In spite of its accuracy, it is needed to have a wider approach, taking into account more than one feature to give a broad vision of the current situation. The aim of our study has been to cluster Listeria isolates according to the tolerance to benzalkonium chloride, the biofilm three-dimensional structure, and the transference by contact to a solid food matrix by using principal component analysis. And why the PCA? PCA decreases the number of the variables in a study system, grouping them into new variables or dimensions. This tool is very useful in food safety risk assessment as it sets and simplifies a wider number of variables into a few dimensions, making the most of the study of the bacterial pathogen in the food chain. The isolates were provided by the EMCC collection and from the Food Technology and Science Department of the University of Córdoba. PCR serogroup was performed following the OFMID protocol, and the benzalkonium chloride resistance was assessed following minimum inhibitory concentration and minimum biofilm eradication concentration in accordance with the classical microbiological protocols. The three-dimensional structure was carried out following a confocal laser scanning microscopy analysis, and the imaging staff were processed using Constat software. The determined parameters were the biovolume, surface area, surface to biovolume ratio, average and maximum thickness, and the roughness coefficient. Finally, we tested cell transference by contact from biofilms on stainless steel coupons to a food matrix, specifically smoked salmon. The results were expressed in percentage of transference in the first 3 contacts. And as a result, 11 variables and 22 isolates were included in the PCA analysis, which was conducted using the FactorMine R plugin and RStudio. From the PCA output, the explained variance was closely to 8%, following the first three dimensions, and the cos squares values show the weight of each variable in each dimension. Thus, the first dimension is depicted by structure parameters such as biovolume or thickness, while the second one relies on the percentage of transference and the surface area. From these scores, a correlation circle is built up. In the correlation graph, the vertical length shows the contribution of each variable to the total, while the position displays the grade of correlation among variables. Taking this into account, we can observe that for benzalkonium chloride, MIC, and MBAC, lack of correlation between each other, which underlines the relevance of the biofilm matrix structure to hinder the disinfectant effectiveness. Also, the MBNC correlates positively with rugosity, which would be accounted for the hindering of the disinfectant diffusion across the biofilm. On the contrary, it has a negative correlation with surface area and biovolume. In third place, the percentage of transference have a negative correlation with the thickness. And this could be explained as the thicker the biofilm is, the higher number of cells could be in the inner layer, ergo less active and incapable of being transferred. Another output was the factor map, which allowed the isolates to be classified in 3 statistically different clusters. No significant correlation was found between the cluster definition and the PCR cell groups. And the layout mirrors a population distribution in which the majority of the strains stands in the middle of the distribution of the first dimension. In addition, by overlapping the correlation circle, we can observe the contribution of each variable to the clusters, as well as showing what variables define better each isolate. Thus, cluster 1 is depicted by biofilm tolerance to benzalkonium, heterogeneity, and low values of surface area and biovolume. Cluster 2 is depicted either by transference percentage or biovolume and surface area. And finally, cluster 3 is characterized by quantonic tolerance to benzalkonium chloride, the thickness, and low values of the percentage of transference. It is also really important to remark that the factor map represents a multidimensional space capturing just 2 dimensions. And for that reason, the isolates located in the middle of the distribution have no representative variable at all, as all weigh the same in the dimension. To summarize, PCA can provide significant advantages for the microbiological risk assessment, considering that it allows to simplify data display and points out the variable correlation. Also acts as an open system, allowing the input of a large number of variables and isolates. But also, in the particular case of Listeria, it provides an accurate picture about isolates distribution, helping to deepen the knowledge in the bacteria population and features. The cluster characterization in terms of predominant variables pre-selects the isolates more suitable for the study of a particular risk trait, as disinfectant tolerance or third transference, making easy the accuracy of future works in food risk assessment associated with Listeria traits. At last, the simplicity and reliability of the PCA makes possible an integrative approach to risk assessment, leaving the door open for future analysis. And that's all. Thank you very much for your attention.
Thank you very much. Do you have any questions from the audience, please? Yes.
In one of your introductory slides, you list some characteristics of Listeria, one being low water activity. All other conditions being optimum, what is the lowest water activity at which Listeria will grow?
Thank you very much for your question. The truth is, I don't really know the answer, because my knowledge about Listeria biological features is very thin right now. So I'm so sorry if I can't answer you right now, but I hope I will be answering you in the future if you are so kind to contact me.
Thank you.
Okay, if there are no other questions, we can move to the next presentation. Thank you very much. It's Mr. Omer Sambach from
The first one.
Israel.
From the Technion in Israel. Okay.
You may
So, thank you for the introduction. My name is Omer Sabach from the Technion, and I'm here today to present my study regarding the microbial community's interactions in sourdough starters using a genome-scale metabolic modeling. So, to start from the beginning, we need to start from sourdough bread. It is a staple food for human nutrition that made much earlier to the discovery of microorganisms. It is composed of 4 main ingredients: flour, water, salt, and of course the sourdough starter. If we will take a closer look inside the sourdough starter, we will see that it is composed of 2 main ingredients, the flour and the water, that are mixed together and then let to ferment. After a while, it will achieve a stable microbial community composed of both yeast and bacteria, mostly lactic acid bacteria. Now, from sourdough starter to sourdough bread, among some other technical parameters, we have the metabolic interactions that are responsible for the bread flavor, aroma, volume, acidity, and much more. But how much do we really know about the exact metabolites exchanges between the bacteria and the yeast, and also among the bacteria themselves? The answer is that we don't know much. And basically, we can refer to it also as a metabolic black box. And that was exactly the aim of our study, to shed some light into this black box. We chose to do it using a genome-scale metabolic modeling, which is a way to represent the cellular biochemistry in a mathematical formal way. In more simple words, we need to take a DNA sequence, a whole genome DNA sequence, convert it into a system of reactions, enzymes, and metabolites that can be illustrated in a nice way, or also in the mathematical formal way of matrix as a system. We solve the system using linear optimizations that actually maximize the objective function of the system. Now, since we are talking biology, the objective of any living cell is to maximize its growth. And this is also the case here in the models. So the first step in our research method was to build the bacterial panel. Here you can see the panel composed of 10 of the most common lactic acid bacteria that occur in sourdough starters worldwide. We build in silico communities, each one composed of one yeast, Saccharomyces cerevisiae, and 3 lactic acid bacteria from our panel. So if we will do it, we will end up with 120 communities to model, and this is exactly what we did. We choose the one yeast and 3 bacteria species to resemble a real sourdough starter composition. So we look at the data, and the first thing that we notice is a correlation between the bacterial community members to the growth rate of the yeast Saccharomyces cerevisiae. Here in the heat map, each row represents a bacterial species, and each column represents one modeled community. So by default, in each column, we have only 3 colored spots, and the color represents the growth rate of the yeast. Now, you can see the diagonal trend pointing to the Pediococcus species and to Lactobacillus sacchae in a way that correlates the percents of those species to enhancing the growth rate of the yeast Saccharomyces cerevisiae. If we will do another analysis, this time the color represents the community CO2 secretion flux. Again, we have here the diagonal pattern showing that the Pediococcus species and Lactobacillus sacchae also correlated to a better leavening ability of the dough. So we ask why, what is so special about the Pediococcus and about Lactobacillus sacchae, and more specifically we ask, can similarity in the consumed metabolites among community members outline a competitive relationships? To answer this question, we use the consumed metabolites profile of each of analysis using a principal component analysis, PCA, of each one of the community members. So in green, you can see the cluster of Saccharomyces cerevisiae, and right on the other side of the main principal component, you can see in shades of orange the clusters of Lactobacillus sacchae and the Pediococcus species indicate that they don't share the same consumed metabolites and they don't compete on the same nutrients. However, the other 6 lactic acid bacteria in our model cluster pretty much above the cluster of Saccharomyces cerevisiae in a way that indicate a positive competition on the same nutrients. So to answer this question, we know that a significant difference in the consumed metabolites profile benefits the microbial community through non-competitive interactions, and of course, vice versa. The other question was, do food chain interactions shape the microbial community properties in sourdough starters? Now, when we talk about food chain interactions, we're looking for a metabolite that's secreted by the bacteria and are later consumed by the yeast. And when we looked on the model and searched for such metabolites, this is the network analysis that we've done. And for example, you can see that the Pediococcus parvulus is secreted histidine to the medium, and the histidine is later consumed by the yeast. And if we look on all of these metabolites, most of them are actually amino acids that are secreted by the bacteria and consumed by the yeast. So we went back to the lab and we did show that if we add amino acids to a defined growth media, it is support the yeast growth in terms to a defined media without any nitrogen source. However, you can see that both histidine and lysine are actually inhibit the yeast growth. Now that was done on the laboratory strain S288C. However, we also expanded it to include some more Saccharomyces cerevisiae sourdough isolates that we have in our lab, also Kazakhstania humilis isolates that we have in our lab, and also to some other commercial baking strains. And all the time we saw the same trend. And once again, lysine is actually inhibit the growth yeast. So we went again back to the models and we classified the bacterial panel into two groups. One group indicated in blue as an average of a positive lysine flux, meaning they tend to secrete lysine to the media. And the other group, here you can see them in red, as an average negative lysine flux, meaning they tend to uptake lysine from the media. And when we did a co-culture assay in our lab, it is actually support the classification. You can see in gray the monoculture of only Saccharomyces cerevisiae alone in a defined media supplemented with lysine. In blue, you can see the co-culture of Saccharomyces cerevisiae and one of the bacteria that was predicted to secrete lysine to the media. We have no difference. And on red, you can see co-culture of the yeast Saccharomyces cerevisiae with one of the bacteria that was predicted to consume lysine from the media and reduce lysine concentration. And when it's happened, we can see that the Saccharomyces cerevisiae is now able to grow to a much count, to a higher CFU to ml. So to sum this up, we know that the bacteria influence the yeast growth also by altering the amino acid flux. But what about the other interaction, the other direction? How is the yeast support the growth of the bacterial growth? So what you can see here is all of the metabolites that we detected that are secreted by the yeast to the media and later consumed by the bacteria. The y-axis represents the Pearson correlation between the bacterial growth to the bacterial uptake flux of the specific metabolites. And we can notice that in red we have amino acid that we weren't detected earlier. In green we have B vitamins, riboflavin, B2 vitamin, and also B5 and B1 that's secreted by the yeast to the media. And we also have in blue some carbon sources such as glycerol and L-malat. So if we have the full picture on these interactions, we can conclude that we have a bidirectional nutrient dependencies between the bacteria and the yeast in sourdough starters. Now the last, and honestly the most challenging question was, can the model's findings help us to predict an actual sourdough behavior? So to answer this question, we took all of this is the CO2 secretion analysis, and we divided all of the 120 communities into two groups. One group that is predicted to have a high CO2 secretion rate, and the other that was predicted to have a low CO2 secretion rate. We randomly choose four communities from each group, and then we synthetically build them in our lab. We inoculated sterile flour and sterile water with the specific bacteria and the yeast, and let it ferment for over 1 week with daily feedings, daily refreshments, just like you make a sourdough. And at the final day, we did our analysis. Here you can see some photo from the experimental setup. You can see how we grow the sourdough starters in our lab, and also the gas production system, where we put in a sealed glass bottle leavened dough made with a specific sourdough starter and let it ferment in a controlled environment for over 20 hours. And we have a success in predicting the CO2 production rate. You can see it both over time, where we can distinguish between the red line that resembled the group that was predicted to achieve a higher CO2 secretion rate. It did achieve a high gas production rate. And we can see the rates in the box plot where we have a significant difference of 20% in the means, meaning that the green blocks plot, represent the communities that were predicted to achieve a high CO2 secretion rate, indeed achieve a higher gas production rate compared to the orange box plot. So to sum it up, if we started from the black box, we now shed some light into it. We know that if we have a similar consumed metabolites, it indicates a competition, and of course the other way around. We demonstrated that we have in sourdough starter bidirectional nutrient dependencies that help to shape the microbial community. And we also showed some in vivo experiments that support the model's findings regarding the Levanian grades. Now, last slide. During the study, my PI always asked me, what about the taste? And at the end of the day, people should eat this bread. So I have here only the preliminary results regarding 30 of the most common aroma and taste compounds that appear to be in sourdough starter and sourdough bread. You can see in the upper green heat map the relative flux, and at the bottom you can see the community member by per column, per community, and we can see some relationship between the metabolites. But still, once again, it is the preliminary result, and we are going to work on it more. To finish, I would like to thank my PI, Professor August Skalkotas, also to Dr. Shiri Freilich from the Volcani Center, to all of our lab members, and for you, for listening. Thank you.
Thank you very much for the presentation. Are there any questions? If there are any questions, now. We have a question from the audience.
If not, is there no? I can't see it. Oh yeah. Okay.
Thank you very much for the very interesting talk. I have two questions for you. The first is, I couldn't understand the first PCA graph you showed, if it was the result of your in silico predictions or was it experimental predictions?
So it was the result of the in silico modeling, yeah.
Okay, okay. And the second question is, why do you have a mechanistical perspective of the influence of so many amino acids produced by lactic acid bacteria in the medium that may help Cerevisiae to grow? Why is lysine, for example, shutting down the growth of Cerevisiae, while the other amino acids stimulate the growth? So
specifically to lysine, there is, it is a well-studied amino acid that we know that impact in a negative way Saccharomyces cerevisiae. The exact mechanism for me it is unknown. I read some articles that suspect it was some byproduct of the degradation of lysine. Some other suspect it was the lysine itself that has a toxic effect on the yeast. But once again, I don't have a specific answer for the mechanism. Thank you.
Okay, thank you very much. We may now proceed with our next presentation, which is going to be via Zoom. Okay, it's by Beatrix Stacey from the University of Veterinary Medicine, Vienna. Yes. Hello. Hello, can you hear us?
Yes.
Okay.
Hello from Vienna.
Hello.
Now back to the Listeria topic. I would like to show you some post-outbreak management data. And we had a problem with a meat processing facility in Austria related to a food outbreak, to a human cluster. And we have the question, when could food production be considered under control again? Sorry, I have a problem with the slides. Outbreak situation, you can also read it by Piecka, which was published in 2019. The outbreak related to meat products, first initially 7 patients got ill from November 2015 to September 2017. And the SNP analysis, so the genetic relatedness between these isolates, differed only by 1 to 4 alleles, genetic alleles. Food and environmental isolates from a huge sort of companies, of Austrian companies, was collected by the reference laboratory, and they compared everything to the human cluster, to this cluster outbreak isolates, and they identified one company, let's call it Company A, where they identified a genetic relatedness, and there were only 0 to 7 alleles difference. And at the end of this whole comparison, they identified the industrial slicer as a source of recontamination. So in meat industry, most equipment which is related to recontamination are slicers. And there were also, in the whole comparison, were also other companies included, also isolates from fish companies and other ready-to-eat products, and also from deer meat products, but they were not considered to be related. The analysis included one scheme applied in Austria, developed by my colleague Werner Rupic. You can also read it within this article, and there is not the whole genome included by this comparison between European reference laboratories. It's only 1,700 genes included from the core genome. The standard process is to compare it by software and to get a minimum spanning tree, what you see in the middle of the slide. And there is the comparison of human isolates from these outbreak isolates and also from the different companies. And the scheme, Originum MLSD scheme by Rupic, identified one clonal type, CT. I marked it in green. It was CT1234, which is corresponding to the Institut Pasteur scheme, and this clonal type calls 100-117. In Austria, we have only this data for the human comparison and also the food-related isolates with this scheme. And when we go to the Pasteur scheme to get a hint how, on a global level, this type connected to clonal complex 155 is present, then we can have a look at the database. And if you have a snapshot on this clonal type, which was related to this human outbreak, you see that the Pasteur site includes isolates from different countries, and the source was, for instance, wild animals, wild pig, deer, tonsils, and also the pork processing facility in one country in Spain. So with this type, wild animals and pork processing environment is related or was related in the past. The genetic type, clonal type, has special features, strain features. For instance, adaptive element, stress survival 1. This is you can find it under attributes, and also has a special adaptation to disinfectants applied in processing facilities. So our situation was a post-outbreak scenario. So with the company, which was then related to the outbreak, 4 food lots were identified positive for Listeria monocytogenes, and different genotypes were already at that situation identified. At the onset of our investigation, the outbreak was already traced back, and there were no human outbreaks for stakes could be attributed to from 2018. We had a critical question. We had a lot of interviews or talks to the management team and also to the agency, And we saw that we all had a critical question that how could a food business operator manage such a post-outbreak situation if he is not aware of its role in the outbreak, and also to work with that whole genome sequencing data. The FBO tried really to manage the situation very actively, so however it became clear that there is in Austria, it's not an authority-approved guideline for the management of a listeria contamination after this outbreak. So the agency decided that if the company is getting rid of all the contamination and if active batch release is applied, that each batch is controlled before it is set on the in the market at retail level, and that this is negative tested for Listeria monocytogenes, then the processing facility might be active with producing food lots. So we started together in 2018 environmental sampling and also tried to map it on transmission maps together with Company A. The processing facility processed different kind of ready-to-eat products, but also convenience products, which, for instance, schnitzel, meat products which have to be heated at the, for instance, at home, and so that it's not ready to eat. So different kind of meat products were processed in this processing facility, and you see that there is a blue line where you can follow the uncooked products, and the red is the ready to eat. At the beginning of the outbreak, only the slicer was identified that this should be related to the outbreak. But then we established a sampling plan and tried to get the hint of the entrance of Listeria monocytogenes and to get rid of Listeria monocytogenes from the environment. If you have a look at the table, we identified the outbreak strain still in 2018. And these are 19 sampling events, and ST155 was present until sampling 7. The general Listeria occurrence in all the environmental samples was 2.4 for Listeria monocytogenes, so it got better through the time. If we have a look at the genetic diversity, in green you see the outbreak-related clones, so it's clonal complex 155 or clonal type 1, 2, 3, 4, and this was detectable at different kind of sampling sites, for instance the hygiene lock, different sites of the hygiene lock, the hallway, then also main locker on the shoes, so it was really widely distributed. Also in the raw material tumbler site, at the schnitzel site, so it was not only identified at the slicer. So we tried really hard to reduce this contamination. And we also identified other genetic types, clonal types, at different sites of the convenience production line and also the raw meat cutting site. So this is the diversity in different colors. You see different genotypes. I will not go into detail with that. So you see the heterogeneity of all the isolates present in the meat processing facility. And then you see the minimum spanning tree. So we identified through these years 4 different clusters. Then one major cluster was identified at the beginning, 2019, and then we got a lot of isolates of clonal type 9, which is very present in pink, and this is very present at meat processing facilities, and it is strongly related to the raw material. So every day, if you work with different meat types, Listeria is entering meat processing facilities at small amounts. So we have to solve the problem at the raw material side. We had some management points, how we reduced Listeria prevalence with an easy way. So we identified gaps, hygiene gaps, and so we saw a lot of isolates from the hygiene log, so we repaired together with the company the hygiene log. This is often a hygienic gap, because this is sometimes the disinfectant does not really get into the hygiene log. Then again, we saw that the slicer team has to be one team, not different persons who are maintaining the whole hygiene standard of the slicer. So at the slicer, we did not identify any Listeria from 2019 on. Then we had to work further. So we saw that conveyor belts were not easy to clean. So we tried to set the sampling plan that we always had the information before and after cleaning and disinfection, that we had a comparison if Listeria is in some niches present still after sanitation. So then we also identified problems with the conveyor belts, and we tried to interrupt some transmission, Listeria monocytogenes transmission cycles occurring between raw material, intermediate product, and slicer. We also saw that the crate washing system was not working very well. The temperature was not fitting. It has to be about 80 degrees, but it only achieved 50 degrees for the main washing with sanitizers and so on. So I will not bother you, but these are some easy ways to interrupt heavy contamination in a meat processing facility. And we have to, this is our outlook, but we really have to go into detail. It is important to track genotypes at the processing facilities, but many types are really entering from the slaughterhouse and also the primary production, so we have to source attribute these types which are very present in the primary processing. So for instance, we get the hint that clonal type 9 and 121 is coming from, for instance, pork producing facilities, as you see here, published by colleagues. But this is applicable for France, but we also have to know this for Austria if it's the same situation, and to compare this then with our colleagues from the reference labs at other countries. Okay, so the outlook is also to go into detail with the adaptation of these strains to the sanitizers and disinfectants, and also to compare it with, to share alternative monitoring concepts, to make tailor-made sampling plans for each company, because in these small and medium small scale enterprises, it does not fit to each company size what you think about monitoring is fitting. So this is the plan. Thank you for your attention.
Thank you very much for the presentation. Is there any question from the audience? Yes, please.
Thank you very much for the presentation. Looking at all the things you found at the production site, it doesn't look like a new and modern and very well-kept production site. So my question would be, were they certified to any food safety standard?
The company, yes, of course. They have IFS audits, and they are controlled, and they have also to, in Austria, they have to sample food lots and also slicer dust, for instance, for meat products, and to verify that they are negative for Listeria monocytogenes. They also have to show that they have a hygienic situation for personnel and also for the processing facility, and they provide those data, but still they get a huge diversity in Austria. They buy a lot of raw materials. So, for instance, this company is diverse. They have poultry meat, pork meat, and beef meat, and this is daily bought by different slaughterhouses. So there's a high diversity entering of Listeria entering this company.
Okay, thank you very much again.
Okay. Thank you.
We may move to our last presentation from the chairperson, Dr. Marion Wells-Benning from Ninso, the Netherlands.
Yes, hello. Once again, good afternoon. I'm going to talk today about an organism that is, by most of you probably known as a spoilage organism and not so much a foodborne hazard. But despite its general profile, we did look at its toxinogenic potential, and I'm going to talk about the spore former Bacillus licheniformis. Let me see how I do this. Yep. So just a very short recap on foodborne illness. So most of you know the pathogens like Salmonella, Listeria, Campylobacter, and these are typically causing foodborne infections. And you probably also all know the more typical foodborne intoxication causing agents, such as cereulide produced by Bacillus cereus, botulinum toxin produced by Clostridium botulinum, and Staphylococcus enterotoxin. However, I'm going to talk today about some less well-known toxins, and particularly produced by Bacillus species. So when I talk about these toxins, it's going to exclude Bacillus cereus. And what I would like to highlight is that, particularly in relation to Bacillus licheniformis, there have been a number of outbreaks reported over the years. Not many, but it is actually happening. And there are a couple of cases where an organism, Bacillus licheniformis, was isolated from infant food and raw milk. Also from contaminated milk powder in a kindergarten, and this actually led, sadly, to the death of a child. And there are other instances where this organism has been found in products that have led to foodborne illness, so mostly the vomiting type. Now, just a little bit on Bacillus licheniformis. It's a spore-forming organism. So as you all may be aware, the organism is growing, and once it reaches a very high cell density, or when it senses nutrient deprivation, it tends to convert, or the mother cell tends to have an asymmetric cell division, and endospores are produced. And you see them here on this little movie, which was made by my colleague Robin Eilander. And once these spores are produced, they are super stable. So this is a good thing to remember. So the spores can be dormant. They can be dormant for even many years or decades even. And not until they experience very favorable conditions, and usually this is due to nutrients, they germinate. So they go from phase bright to phase dark. And then from this phase dark, spore cell emerges, and then growth can reappear. So this is just a very short recap again on sporulation and germination. And when we look at Bacillus licheniformis, it is an organism that is ubiquitously present, so you find it everywhere really. So in decaying plant material, in soil, in water. And it is quite a robust organism. It can grow at a very broad temperature range from, let's say, 7 degrees up to 60. And when you're culturing it, it's actually sometimes very difficult to handle on plates. It's a slime former. It spreads very easily. It can overgrow plates. And actually this ability to to spread on plates and to swarm is very likely related to the compound that is actually also causing foodborne illness. And that is the compound, lichenisin. So lichenisin is very similar to another compound produced by Bacillus subtilis, and it's actually, the structure is shown here. So these compounds are surfactants. They have a very potent ability to reduce the surface tension, of where they are growing, essentially. So these compounds, they are produced, they're non-ribosomally synthesized, so it's a very large gene cluster that produces different modules, and these modules actually couple different amino acids, and then there is this circular peptide has a carbon tail, which typically is around 15 carbons. So this structure is extremely stable. And this is actually part of the problem for food safety. So you can actually sterilize it, nothing will happen to it, it's very stable. So what is known about this compound is that it causes membrane permeabilization, and that in turn causes nausea and vomiting. And some known concentrations needed to establish that have been established around 40 to 60 micromolar. Now, Bacillus licheniformis, a step back to food, it's a very common contaminant in food ingredients, and it's been quite well known that in milk powders, it can be the predominant species. So it really depends on the processing facilities. So in milk powders, you can get very dominant presence of very high heat-resistant spores like Geobacillus, but depending on the location and the facilities where milk powders are produced, it can also be Bacillus licheniformis, and there's quite a few studies on that. So coming from the background at NISO on milk powders and dairy products, we have actually been transitioning to plant-based materials and being able to make plant-based dairy alternatives. So we were actually trying to make all these products, and lo and behold, we were encountering Bacillus licheniformis in a lot of the products we were trying to make. So we were actually wondering, okay, what about this contaminant in all these plant-based sources? So we started to do a survey on a lot of different compounds like pea and fava bean and chickpea, almond, and what we actually found is that the majority of all these ingredients, they contain quite high levels, sometimes up 10 to the 3, 10 to the 4 spores per gram, and of spore formers, and nearly all of these samples contain Bacillus licheniformis. And maybe this is not very surprising considering its natural amnesia, so it's really associated with plants and perhaps also root systems. But this is definitely something to bear in mind when you're dealing with these type of new ingredients. What is worth mentioning, other than that it's common in foods, some of the spores are very difficult to kill. So usually licheniformis spores will be killed by UHT, but 10% of all the strains that we've been isolating contain genetic elements that are actually making them more heat resistant. And quite a few, so roughly around 10% of all these strains, you cannot inactivate them even by boiling them for half an hour. And this is actually the type of heat that, is really the max for what you can use in plant-based ingredients. So then, coming to the aim of the study, so knowing that this organism is present in a lot of these plant-based ingredients and the type of products we want to make with these, where you cannot apply very high heat loads because it actually destroys the properties of the products we would like to make, we wanted to assess what the risk was of lichenisin production in foods. We have actually done quite a bit of work on plant-based ingredients, but I'm not presenting that today, but I would like to present the work that we did on the initial work on LB medium and on skim milk. So what we did, we took 11 different food isolates of Bacillus licheniformis, and we used these to culture in LB and milk, and we assessed lichenisin production And in addition, we looked at the toxicity to intestinal cells to see, okay, you know, the levels that you may encounter in food, could that be relevant to what you can experience for human intestinal cells? Well, this is a very detailed overview. It's actually in the publication that we have. I'm not going to go through this in great detail, but what we did here, we took 11 real food isolates, so you can find them anywhere really, but these have been problematic in foods. And in addition, we took a type strain, DSM 13, and we established just its common, or its growth potential, looking at growth at different temperatures, presence of salt, ethanol, and also establishing whether or not it could grow under anaerobic conditions. And what we saw is that the growth of the organism is actually really robust. It grows at 37, but most of the strains also grow very well at 55 degrees. They grow well at 37 and 55, even in the presence of 1 molar of sodium chloride. The organism grows in the presence of 4% ethanol and under both aerobic and anaerobic conditions, in this case actually supplemented with some potassium nitrate. In addition, we looked at the genetic profiles of these strains, and we did find these lichenisin clusters in there. And in addition, we established whether they could produce the lichenisin, and we found that was positive for all the strains. But some were much better than others, and this is just displaying for the situation for 3 strains. So on the left here, you see this reference strain, the DSM 13 strain, and the 2 other strains are quite high producers. In the top, you see in the red plates are hemolysis caused by these organisms, and you see that these 2 strains on the left are actually lysing blood cells. And in the bottom, you see an oil displacement. So what you actually do is you drop some of the supernatant onto oil, and if it has this ability of this detergent ability, it displaces the oil, and then you see the water underneath. And actually this was very apparent for some strains, especially for these very strong producers. So we carried on the rest of the work with these strains. And what you see here is actually the growth of these strains in LB and in skim milk. So LB on the left, skim milk on the right. And you see that it grew faster in LB than in skim milk. But in both cases, very high cell densities were reached, around 10 to the 9. But if you then look at the ability to produce lichenisin, this was quite substantially different in LB versus skim, so much higher concentrations in LB, and in addition, lower concentrations in skim milk. But what you also see is that, so the white strain is a relatively high producer, the black bar is likewise, but this reference strain, the DSM 13, was very poor producer. So this actually indicates that there is a very large difference between the strains. I'm not going to go in very great detail on all the things we did. We also looked at different chain lengths, etc., and they may actually have effect on the toxicity of the compound, but that can all be found in the paper. But one other feature I would like to highlight is that the temperature at which the strains were grown was also quite determining in the levels of lichenisin produced. And this is shown here, and we saw actually that the strains, when they were grown at 55 on the right, they produced less lichenisin than at the temperature of 37 degrees. Just to get back to the previous slide, so the levels that we found were actually around 36, let me see, how much was it? Yeah, 36 microgram per milliliter. And this now relates to the levels that, this was produced in the liquid medium. And we also assessed what the concentration of lichenisin was to reduce the cell viability, in this case of CACO2 cells, by 50%. And we found that this was in the range of, let's say, 17 microgram per milliliter. So what is really important, I think, as a take-home message is that lichenisin can be produced at levels that are physiologically relevant. Of course, you cannot make a direct relation between how much is present in a drink and how much is having an IC50 in the human gut. Of course, there is the ingestion and whatever happens in between, but the levels are definitely relevant. And that's obviously also clear from the outbreaks that have occurred in the past. One last thing that I would like to remark is that the levels that the moment you start to see lichenisin production is really in the late stages of growth. So it's the late exponential phase and the stationary phase. This is where you see lichenisin produced, and we never saw it under the level of 10 to the 5 CFU per mil. So if Bacillus licheniformis is present in food at very low levels, under 10 to the 5, let's say, it's very unlikely that this will pose a foodborne hazard related to lichenisin production. However, there's always this potential of the risk of foodborne intoxication by this organism, and that really depends on the strain that is present, the food composition, and the storage condition of the food. So, yeah, to keep a product safe, it's either one of the two. One is to inactivate all the spores, make sure they're not there. The other would be to prevent outgrowth. So with that, I would like to conclude my presentation. I would like to thank all the co-authors on this. So Claire Yeek, she's nearly at the stage of defending her thesis next month. Manza, Guido, they were actually part of the study at NISO. Blanca and Jerry at the I see Chocolate, the host microbe interactomics group in Wageningen, and Chaco Abe himself is also co-promoter of, promoter of Claire. So thank you very much.
Thank you very much for this nice presentation. Are there any questions from the audience? Yes, please.
Hi.
Very nice talk, Marianne. Good to see you again. I had a question. I was just wondering, can you consider lichenisin a kind of a variant of surfactant?
Not quite. It has two very distinct differences in the amino acid composition of the ring.
Yes.
And that is pretty consistent. So.
Oh, okay.
But in a way, yes.
And is their cytotoxicity the same?
It's very similar. It's very close to each other. Yeah, we did test the surfactant as well. I left it out of this presentation.
There was, I don't know, maybe you've seen it, there was a few publications on anti-COVID activities of surfactant and these, because, of the lipid outlet that and so they're anti-COVID, I don't know.
I'm sure it will be anti a lot of things.
Yes, it will be, but I'm sure any detergent will be probably,
yes.
But in these organisms, a lot of these organisms, like you have velinesensis, amyloliquefacients, they produce a lot of these kind of thiopeptides, including fengisin and also bacteriocins like amylocyclicin. I was just wondering, in the repertoire that's in licheniformis, what do they produce? Do they produce fengisin,
licheniformis not so much. Subtilis has a bigger repertoire than licheniformis. But what is very striking is that all strains contain, as far as I've looked, all strains of licheniformis contain this non-ribosomally synthesized, this cluster, which is huge. So it's a very big genetic burden on the strain. So it must have a very big advantage for the strains to be able to produce it.
Thank you.
Okay, so I think we're, I don't know if anyone else has questions, please come and see me. I think we're ready for coffee. So, or if it's there. Or or the end of the session.